Hello, everyone. My name is Shanling, and I am the study director from the Neurological Disease Pipeline in GemPharmatech. Very glad to be here to introduce the mouse models and the preclinical surveys for neurological disease research in GemPharmatech to you. Firstly, let me do a brief introduction of our company, GemPharmatech. GemPharmatech is a global biotech company dedicated to providing a one-stop solution for biomedical research using genetically engineered mouse models. Our co-R&D and management team has over 21 years of experience in mouse model development, and we have been granted over 40 technology patents and maintaining a stock of over 22,000 mouse models, generating over 6,000 new models annually. And this makes GemPharmatech one of the largest mouse model supplies in the world. In addition, GemPharmatech is more than just a model supply. We also provide a one-stop solution for your research needs. We offer a wide range of mouse models and service, including those for the neurological disease, immuno-oncology, metabolic disease, autoimmune disease, rare disease, and so on. And our services are provided through the different platforms like the flow cytometry platform, phenotyping platform, behavior testing platform, and the PK or PD service platform. And our preclinical service platform undertake over 1,000 preclinical efficacy studies annually, and more than 200 R&D applications for NMPA and FDA. Next, let's move on to today's main topic, neurological disorders. The concept of neurological disorders is very broad, encompassing the disease that affect the central nervous system and the peripheral nervous system. Therefore, the types of the neurological disorders are very diverse, including the neurodevelopmental disorders, neurodegenerative disorders, and other diseases that affect the nervous system. According to a new study published in the Lancet Neurology in March of this year, by 2021, more than 3 billion people worldwide were affected by neurological disorders. And this accounts for approximately 43% of the world population. And this figure making the neurological disorders the leading cause of illness and disabilities in the world. The mechanism of neurological disorders are very complex and involve multiple factors. The genetic factors play a crucial role in in many neurological disorders. The conditions like Huntington's disease or amyotrophic lateral sclerosis have been confirmed to be caused by the genetic mutations. And the environmental factors are also significant. For example, the cardiovascular disease are often linked to the chronic conditions like the hypertension or the diabetes. And additionally, the immune system abnormalities can lead to the neurological disorders, like the multiple sclerosis, which occurs due to the immune systems affecting the nerve fibers. Due to the large patient population and high market demand, the drug market for the neurological disorders has experienced a steady but moderate growth, mainly because of the complex mechanism of this disease. And according to the Dimension Market Research, the global CNS drug market is projected to reach 66.4 billion by 2024, and then further growth to over 100 billion by 2033, with a compound annual growth rate of 5.1%. However, compared to the diseases like oncology disease or autoimmune disease, the R&D barriers in the CNS field are high. Some small breakthroughs can potentially lead to the longer product life cycles. And also, lack of some new targets over the years has increased the investment value of modified some new drugs that could address the unmet clinical needs. That's the reason why some big pharmas continues to enter this challenging market. So to support the mechanism research and the drug development for neurological disorders, GemPharmatech has independently developed a series of mouse models related to this disease. Currently, our platform is divided into the following four main sections, including the neurodegenerative disease, pain disease mouse models, the psychiatric disease mouse models, and mainly the CUMS-induced depression-like mouse models. And the other disease like the neurological related the rare disease, including the amyotrophic lateral sclerosis and Huntington's disease. For clinically common neurodegenerative disease like Alzheimer's disease and Parkinson's disease, or the Huntington's disease and amyotrophic lateral sclerosis, we all have one or more mouse models that are available for study. For example, in the case of Alzheimer's disease, we already have FAD2T, FAD3T, and FAD4T. These three models that are available in the market. And the different numbers here represent the different counts of mutations carried by each model. In addition to the gene editor models, we also offer virus drugs or surgery-induced disease mouse models. For example, the pain disease mouse models and the depression disease mouse models we just mentioned are constructed through the induction method. The mouse models marked in orange here are already available, while the others are still under development. And I will provide a brief introduction of these available mouse models later. For each disease mouse model, we typically introduce it from the following aspects: the background of the disease and the hypothesis of the pathogenesis, the construction strategy of our mouse model, and some detailed data, including the behavior data, the pathology analysis, and the efficacy study. And we will also do the comparison with other mouse models that are commonly used in the market and provide you with some relevant application scenarios. Then I will take Alzheimer's disease mouse model as an example to introduce the data we can provide for each disease model. Alzheimer's disease is the most common neurodegenerative disease in the world. And in 1907, the two key histopathological hallmarks of Alzheimer's disease were for the first time described by Dr. Alzheimer when he examined the brain sections of his patient. One is beta amyloid plaques, and another is the phosphorylated tau protein related the neurofibrillary tangles. Mutations in certain genes, such as the amyloid precursor protein gene, APP gene, and the presenilin genes, PSN1 gene, are known to increase the production of beta amyloid or alter the ratio of A-beta-42 with A-beta-40. In the individuals with these genetic mutations are at a high risk of for developing the early onset Alzheimer's disease. So to better understand the disease pathology and to identify the mouse models suitable for investigating the potential therapeutics, we developed these three AD mouse models, FAD2T, FAD3T, and FAD4T. These models target the APP and the PASN1 or the MAPT mutation, gene mutations, respectively. The most commonly used AD mouse model is FAD4T mouse model. FAD4T mice have four AD-linked mutations: the Swedish mutations and the Indiana mutations in human APP gene and the M146L and L286V mutations in human PASN1 gene. Compared with the commonly used mouse models in the market, like the 3xTG or 5DX, which are not available for the commercial use or for the companies, FAD40 mice have independent IP, and that means the cells are not limited and everyone can use it. And then is the detailed pathology and behavior data that we have collected according to the timeline. And as we can see from this figure, the amyloid plaques can be detected in the cortex and the hippocampus as young as 2 months of age and gradually increase with age. And the expression level of A-beta 40 and A-beta 42 protein has shown the same increase. Glysis of the astrocyte, which labeled by GFAP, can be detected in the cortex and hippocampus at 2 months of age and also increase with age. And glysis of some microglia, which labeled by IBA1, can be detected also in the hippocampus and cortex, but from 4 months of age. Synaptic loss, which labeled by the decreased expression of PSD19-5 protein at 10 months of age in FAD40 mice. And to measure the spatial learning and memory of FAD40 mice, we performed the Morris-Water maze test. And for 8 months of age mice, especially the female mice, the female FAD40 mice showed the natural learning and memory abnormality compared with the wild-type mice, indicated by the less crossing number, and they spend less time in the platform area and also in the tagged codons. And some low dose drug from our collaborator can improve the learning and memory ability of FAD40 mice in Morris-Water maze. In addition to the basic model data, we have also collected the efficacy data for the positive drugs in some disease mouse models, either independently by ourselves or through collaborations with other research groups. For FAD40 mouse model, we used donepezil, memantine, and aducanumab, these three positive drugs. And according to the quantification analysis, treatment with these positive drugs could partially rescue the pathological phenotypes in FAD40 mice. And more importantly, we do some comparison with other commonly used mouse models in the market or other AD models in GemPharmatech. For example, compared with the 5DX mouse models from Jackson Lab, our FAD40 mouse models show two obvious advantages. One is the early occurrence of the pathological phenotypes, like the A-beta plaques or the glysis of microglia or the astrocyte. And this will be suitable for the customers who conduct research and the drug development, targeting only the pathological phenotypes. And the other one is the congenitive impairment. Our FAD40 mice showed the congenitive impairment at 8 months of age, which is later than the 5DX mouse models. And this will be suitable for the drug evolution studies, which requires long intervention periods. And here are the articles published using our FAD4T mouse model. In addition to FAD4T, we also have another mouse model called FAD2T, which carrying the human APP and the human Parkinson gene, but with only one mutations in each gene. This model exhibited the mild A-beta symptoms, and we detected the A-beta deposition from 4 months of age. And the total amount of the A-beta is lower than those in the FAD40 mice. But the congenital dysfunction occurred at the same age as the FAD40 mice. Both male mice and female mice exhibited learning and memory abnormality compared with the wild-type mice at 8 months of age. So compared to the FAD40 mouse model, our FAD2T mouse model exhibited later A-beta amyloid aggregation and lower A-beta protein expression levels. So it's characterized by mild to moderate pathological profiles of A-beta plaque accumulation and has a longer therapeutic windows compared with the FAD40 mouse model. So that's making it more suitable for the development of vaccines or some preventive treatment drugs. However, it's important to note that, like all other AD mouse models in the market, none of them could fully replicate the entire spectrum of Alzheimer's disease. So our group is diligently working towards developing a new mouse model that encompasses the full spectrum of Alzheimer's disease, including the progression involving A-beta and the hyperphosphorylated tau. Recently, we have launched a new and advanced AD mouse model called FAD3T mouse model. And this model features the mutations in both APP and the tau-associated gene MAPT. And this model promised to closely mimic the AD from diagnosis to behavior manifestations. And we introduced the human APP and the human Parkinson and human MAPT gene into the mouse genome to construct the FAD3T mouse model. This model was validated and analyzed from the 3 dimensions, the clinical diagnosis, biomarkers, the pathology, and the behavior analysis. Firstly, these are the blood diagnostic biomarkers. In 2023, a draft of the latest AD diagnostic guideline was presented at the Alzheimer's Association International Conference, which included the blood diagnostic biomarkers. The blood biomarkers have shown the high occurrence in AD diagnosis in recent years. And in FAD3T mouse models, we also found that the ratio of A-beta 40 with A-beta 42 is significantly high in FAD3T mouse models at 1 month of age, but with no further increase with age. And compared to the wild-type mice, the expression level of phosphorylated tau 1A1 in FAD3T mice are significantly elevated and gradually increase with age. But the phosphorylated tau 217 levels in FAD3T mouse models show a slight increase at 1 month of age, but there is a significantly increase at 2 months of age. And the levels of neurofilament light chain, the NFL, in the plasma significantly increased at 3 months of age. And this appearance and the sequences of these diagnostic markers in the plasma are consistent with the clinical manifestations of a Alzheimer's disease patient. The pathological changes of AD patients mainly include the deposition of amyloid plaques and the appearance of neurofibrillary tangles, and also the occurrence of some inflammatory response in the brain. So in FAD3T mouse models, we also detected these three pathological markers. Firstly is the A-beta plaques. The A-beta deposition was first detected in the cortex and hippocampus at 2 months of age. And comparing the A-beta levels between 2 months of age, 4 months of age, and 6 months of age, accumulation of A-beta plaques was detected in their brains. And the accumulation also of phosphorylated tau, which is the phosphorylation site in THR1A1 in male and female FAD3T mice starting at 1 month of age, and the aggregation gradually infects with the increasing age. And the accumulation of the phosphorylated tau with the phosphorylation site in SER202 or THR205 also be found in both genders of FAD3T mice from the 3 months of age. And the changes related to inflammatory response can be assessed using the glial cells as indicators, and the numbers of the astrocytes increased in the cortex of FAD3T mice at 6 months of age, and so did the microglia cells. We also performed the Morris-Watermaze test to measure the learning and memory functions. The 3-month-old female FAD3T mice exhibited the memory impairment in the Morris-Watermaze, but no difference in the learning stage, while the male mice began to show both learning and memory abnormalities at 4 months of age. For FAD3T mice, we use lecanemab as a positive control to evaluate its efficacy. Lecanemab is currently a popular drug for treating Alzheimer's disease that targets the amyloid beta and has been approved for clinical use in many countries. And starting from 1 month of age, the FAD3T mice received a weekly injection of this antibody drug for 4 consecutive weeks, and after which the tissue were collected for analysis. And the experiment results here show that the lecanemab reduced the levels of 2-phosphorylated tau protein in the plasma to some extent, but no statistically significant difference were observed. And similarly, the lecanemab reduced the area of A-beta plaques in the brain to some extent, but also no statistically significance was observed. And we will attempt to extend the administration duration of lecanemab to see if we can get a better result. And taken together, our new advanced AD mouse model, FAD3T, not only reflects the full spectrum observed in AD patients, including the diagnostic biomarkers, the pathology, and the behavior changes, but also accelerates the timeline and depth of the mechanism research and drug discovery. The positive control lecanemab show a trend towards improving the pathological phenotypes in the FAD3T mouse model. So please to look forward to more data on positive controls in our subsequent studies. And please stay tuned for update of the FAD3T mouse model from GemPharmatech. And most of the existing AD models now are constructed by introducing the human APP gene mutations, which carried by the familiar AD patients. But the proportion of familiar AD patients in the clinical AD patients is less than 5%, and more than 95% AD patients are sporadic AD. So based on the FAD4T mice and the FAD3T mice, multiple sporadic AD-related risk genes are introduced to better simulate the disease conditions of sporadic AD patients. And we called them the AD plus mouse models. And now we have we hope these models have both the disease phenotypes and the human-sized therapeutic targets. And now we have the FAD40 mouse model crossed with the human TRIM2 target, and more data is being collected in progress. And we also prepare the human TFRC mouse models to help the antibody drug to crossing the blood-brain barrier. So if the drug you are developing is related to this target, so please look forward to our these two mouse models. For the remaining neurological related mouse models, I will focus on the basic phenotypes and the comparisons with other mouse models. If you would like to know more detailed data about these models, so please refer to our strain information or contact with our sales manager. Firstly is the Parkinson's disease. Parkinson's disease is characterized by the accumulation of alpha-synuclein in Lewy bodies, and also with the degeneration of dopaminergic neurons. And for Parkinson's disease, we constructed these three mouse models based on different pathogenic hypotheses. The first one is the T5MCQ mice, which constructed based on the mitochondrial dysfunction. Conditional knockout of the T5 gene in dopaminergic neurons leads to the death of middle brain dopaminergic neuron. And the other two models were related to the accumulation of alpha-synuclein. Different mutations in human SNCA gene destroy the original molecular space and the structure and the stability of the alpha-synuclein protein, and resulting in the fissure of normal degradation of the alpha-synuclein, thereby causing the neurodegeneration. And in these two mouse models, there is one mouse model carrying two mutations and named the E46K-A53T. And this is the unique mouse model developed by our company. Compared to the mouse model with only one mutations in SNCA gene, this mouse model exhibits earlier alpha-synuclein aggregation at a high level, and also exhibited more dopaminergic neuron loss and more severe behavior phenotypes. And the median survival time of these double mutations mouse models was about 1 to 2 months longer than the mouse models with single mutations, showing a longer therapeutic windows for efficacy evolution. Therefore, all the three PD mouse models showed the pathological dopamine neuron loss and also the motor dysfunction consistent with the clinical PD patients. Only the T5MCQ mouse models didn't show the alpha-synuclein aggregation. So if your research focuses on the alpha-synuclein, you can choose the later two mouse models. Another PD mouse models is the 6-OHDA-induced PD models. This model also shows the difficulties in motor coordination and loss of dopamine neurons. And compared with the models in other company, we have improved the success rate of model establishment to about 70% and extend the drug administration windows. Next one is the amyotrophic lateral sclerosis, ALS mouse models. And the two mouse models of ALS were constructed based on the different genes linked to the ALS. One is TLDBP gene and another one is SOD1 gene. Both mouse models show the phenotypes of muscle atrophy, motor neuron loss, and decreased the motor function, and that also occurs in the ALS patients. And these two models are suitable for screening the efficacy of gene therapy or some small nucleotide drugs like ASO, which targeting the specific gene. Compared with the commonly used SOD1 mouse models from Jackson Laboratory, our SOD1 mouse model exhibited the later disease onset and more in the middle age, which consistent with the age of onset that we observed in the clinical patients. And compared with the TDP43 mouse models from Jackson Lab, our TDP 43 mouse models exhibited a longer lifespan. For Huntington's disease mouse models, we only have one mouse model named the B6 human HTT CAG130 mouse model. We transferred into a human HTT gene fragment with carrying 130 CAG repeat expressions to simulate the pathogenesis of human Huntington's disease patients. And for this Huntington's disease mouse models, the greatest strength is a longer lifespan. The course of disease is about 4 months, which will be better for long-term drug efficacy study. And for Duchenne's muscular dystrophy, the DMD disease, and the related gene of DMD is the dystrophin gene. And we have constructed different DMD models based on various exon deletions, such as exon 45, 50, 51, and 52 deletions. And these mouse models were suitable for the exon escaping therapy or the gene therapy for DMD. For PAM models, we have a total of 3 types of PAM model platforms. Inflammatory pain induced by the formalin or CFA, and the neuropathic pain, which constructed by the sciatic nerve crash, and also the neuropathic pain induced by disease or different chemotherapy drugs. And for each PAM model, we have 3 types of assessment indicators, including the animal behavior. We tested for the Von-Free test and the head-cold plate test or the get-analyzed test, and the pathological tests through the skin biopsies and marker detections in the spinal cord or the DRG region. And also, we detected the pain-related cytokines or the chemokines in the blood, which were quantified using some specific method. And we also have other pain models under development, such as the migraine model. And for the recently popular mouse models of psychiatric disorders, we currently have CUMS-induced depression mouse models. And we are also in the process of developing mouse models for other psychiatric disease like the autism or the anxiety disease. As mentioned earlier, in addition to the mouse models, GemPharmatech can also provide the preclinical efficacy service. And all the data we have presented above are constructed on our own preclinical efficacy platform. Here is an overview of the service that we can offer. We can provide a series of preclinical drug efficacy evaluation service or the phenotype screening service of some new mouse models based on our preclinical efficacy platform, including the drug administration, modeling, behavior analysis, in vivo biochemical detection, the sample collection, and the in vitro pathological analysis, and more. Throughout the entire workflow, the first step is the drug administration. And in terms of the drug delivery technology, we not only have the common injection methods like the IP, oral garbage, the RVSC injections, but we can also provide some delivery methods specifically for the central nervous system. For example, the intracranial injection or the intracerebral injection, which can help the drugs cross the blood-brain barrier. And based on the literature or the experience, we can provide the customers with the maximum dosage range for different administration methods. In terms of the behavior analysis, if you are interested in the Parkinson's disease or some other motor disorders, we have a series of motor function testing instruments for you to choose from, like the grip stress test, the rotor test, get an analyze, and the treadmill. And if you are more concerned about the congenitive disease, like the Alzheimer's disease, we have classical, the Morris-Water-Maze test, and the Y-Maze test, and also the novel object recognition. And in addition, we also have the bone-free and the hot cold plate for the pain testing and the tail suspension and forced swimming or the sugar water preference test for the detection of mental disorders like the depression or the anxiety disorders. Taking the Morris-Water-Maze test as an example, it's mainly used to evaluate the spatial learning and the memory ability of the mice, and the following indicators are used to jointly determine whether the mice have congenitive impairment. For example, our FAD mouse models compared with the wild-type mice, our FAD4T mouse model showed the natural learning and memory abnormalities indicated by the less crossing number of platforms and less time stayed in the platform and less time stayed in the target quadrant. And for the in vivo biochemical detection, we can provide the in vivo image platform to detect the drug distribution, and also the DEX to detect the bone mineral density and the body composition, and also the metabolic cage. But these two are more used in the metabolic disease. And for additionally, we can collect the virus tissues after the experiments, such as some specific brain regions samples and the spinal cord or the CSF, which are commonly used in the neurological studies. For in vitro pathological analysis, we can do both the histological analysis and the molecular analysis. For histological analysis, we can provide the chemogenic staining, the immunohistochemistry, and the immunofluorescence studies. And also, we can provide the pathomorphologic evolutions. And for molecular analysis, the gene expression or the protein expression can be detected by the qPCR, Western blot, or the ELISA kit, and we also can provide the blood analysis. And here is an example of the immunohistochemistry showing the beta deposition in the FAD4T mouse model, and the immunofluorescence result showing the astrocyte, which labeled by GFAP in the hippocampus in FAD4T mouse models. That's all the introduction of our neurological disease mouse models and the platform. And in summary, GemPharmatech can not only offers a wealth of model resource, but also we can serve as a reliable partner for your preclinical efficacy study. We are unable to provide all the detailed information about all our models due to the time limits. So if you are interested in any of these models, so please feel free to email us to obtain the detailed data.